Okay, welcome back, guys. We talk about DNA fingerprinting, so now we're going to talk about genetically modified organisms and also what it means to create a genetically modified organism. All right. So what is genetic engineering? So genetic engineering is modifying DNA, which means to change it, and we do this by adding or removing parts of the DNA sequence. It can be used to create what are called transgenic organisms, and these are organisms that have foreign DNA. In this image, we have two transgenic organisms. We have a glowing fish and glow-in-the-dark kitties. So there are fish that naturally glow in the dark, and so they have created a transgenic organism by taking the DNA from the glowing fish and putting it in a kitten to create a glow-in-the-dark cat. All right, so other than just for the fun, why do we create transgenic organisms? All right, so we do this for various reasons, but how do we do it? First, we take what is called a restriction enzyme. Restriction enzymes are scissors, but they can only cut in a certain place. All right, and these restriction enzymes are used to isolate a particular gene. Okay, so for example, we would use a specific restriction enzyme to isolate the insulin gene from human DNA. When we cut it, we have overhang, which are called sticky ends, because they want to bind back together. So we have to use certain things to help those sticky ends from going back together, but we have this overhang. Okay. Then what we would use is the same restriction enzyme to cleave a vector. Okay, so a vector is some way that you're going to put the DNA into something else. So generally, a vector is a bacterial plasmid. Remember, a plasmid is the circular bacteria DNA. So bacteria's chromosome is circular, and it's called a plasmid. Think back to previous units. All right. Simply, if I say vector, a vector is a structure used to carry foreign DNA. All right, so we would use the same restriction enzyme that we use to cut the human insulin DNA to also cut an opening in our plasmid. The sticky ends would then be complementary, and they would come together and bond. And then what you have is called a recombinant plasmid. All right, so we insert the foreign DNA fragment into the vector, and this is called splicing. Splicing is putting them together. All right. This combination is possible for two reasons. Okay. First of all, we know that DNA in all organisms is made up of the same thing: deoxyribose sugar, phosphates, and the 4 bases. Second, restriction enzymes on both samples will work and cut at the same places. So here's a lovely image where we take the restriction enzyme to cut the plasmid, insert human DNA into the circular plasmid. That ends up being recombinant plasmid. We then use the bacteria to infect, all right, a cell to insert that DNA into the cell's original DNA. And that can allow us to do things such as produce insulin in cells where it's not possible. All right. So the combined DNA is called recombinant DNA, is inserted into the host, like I said, which is generally a bacteria cell. The host will then copy and clone. So cloning is the same as copying. It'll copy that DNA, and it will produce the proteins such as insulin. So for this example, we've put the human insulin DNA into a bacterial cell, and it will produce insulin. We can then use that insulin that is produced to give to people who don't produce enough insulin, who have diabetes. All right, so here's another image. We have the plasmid vector, sticky ends. We insert the foreign DNA. We use the enzyme ligase to put it together. Remember, we talked about ligase and DNA replication. Okay, it ligates things together, and then we can use it to transform into a bacterial cell. All right, so what are we using this for? Okay, so it can be used for gene therapy. All right, gene therapy is used to treat various disorders. One of those is severe combined immunodeficiency, also known as bubble boy syndrome. And we also use it for cystic fibrosis. All right, it's safe for use for SCID for up to 10 years. However, it does increase the risk of developing leukemia, but it allows these people to live more normal lives. All right, in people with cystic fibrosis, results have been limited because they are finding that the patient's immune system will often fight off the virus that is used to carry the gene to the target cells. Because you can also use viruses to carry these recombinant DNA vectors to cells that need it. All right, so how does this work? So first you identify defective genes within the DNA sequence. All right, so individuals can be tested for the presence of defective genes. So for example, in the bubble boy syndrome, as we were talking, the defective gene is called IL2RG. All right. After that, a functioning gene is isolated from donor DNA, all right, and this is packaged into the vector, all right, and they usually use something like a cold virus for stuff like the cystic fibrosis gene therapy or other viruses, depending on where they need to get the gene to. Then the vector is introduced to the organism with the defective gene. So this is a very scientific way of saying we infect the human with the cold virus that has the correct gene. All right. What happens is the functioning gene is then delivered to the cells that need it, and it inserts itself into the DNA. Now, the defective cells can now produce the correct protein that is necessary. And this can allow these people to live more normal lives. All right, so there are many practical purposes for genetic imaging. It can also be used to produce large quantities of human proteins, such as insulin and human growth hormone. Both of those are used to treat people. Insulin, of course, people with diabetes. Human growth hormone is used to help people who are not meeting their growth milestones. They're small, and they'll take human growth hormone. And those they use genetic engineering to produce large quantities of these. And it can do so cheaply because they can provide animal models for human genetic diseases also. So when I worked in research, we used what are called knockout mice. We modeled genetic diseases, whether it was prostate cancer, schizophrenia, autism, all those things, by creating mice that didn't have genes or had human genes inserted into them. All right. Agricultural applications are also a big use of genetic engineering, but there's also a lot of controversy around this. Okay, so they can produce plants that are resistant to herbicides or pesticides, such as Roundup resistant soybeans, and these are called GMOs, or genetically modified organisms. Okay, also industries use we create genetically modified organisms to have microorganisms to clean up mining waste, sewage treatment, and environmental disease. So what happens to the water when you flush the toilet? It goes to a sewage treatment plant. It is cleaned using microorganisms that have been genetically modified to do that, and then the water is cleaned and then put back out and used again. So if you want to learn more about that, there is a video on YouTube called The Poop Cycle, and you can look at how the water you flush down the toilet comes back out your faucet later. All right, so there's some bioethical concerns raised by genetic engineering. One is we need to be thinking, should we instead of could we? For example, should we alter the natural variation of human genes by genetic engineering? So also, creating plants with new genes could also cause allergic reactions from unsuspecting consumers. All right. So for example, if you add a gene from a peanut plant to a corn plant, a person who has a peanut allergy may react to that corn plant because they may have been actually allergic to the gene that came from the peanut plant. All right. And also, creating organisms that are not naturally occurring could cause environmental concerns for both just the environment or for humans. So, an oil digesting bacteria may get into oil-based machinery and could also get into our oil supplies. All right. So, the next thing is, how do we know these genes that we're cutting out? How do we know what they are? That comes down to what's called the Human Genome Project. All right, the Human Genome Project was a collaborative effort among scientists around the world to map all the genes in the human genome. It was to identify the location and sequence of all genes on all the chromosomes so that we could better understand human genetics. Um, all right. One of the applications of the Human Genome Project is to determine whether individuals carry the genes for genetic conditions. And this also further allows us to develop gene therapy or genetically based medicines. All right, so there's some serious bioethical questions concerning this. The Human Genome Project allows for the development of genetic screening, and this question is, if we know something's wrong, should the patient be notified? And should insurance companies be able to require genetic testing before they insure people? So that's a huge bioethical question. Next, it has identified the location of genes, and this allows for the idea of designer babies. Could we choose our babies to have brown hair and blue eyes? And also, it allows scientists to determine the genes important to medicines, and they could patent gene sequences for use only within their company, which is a huge ethical debate. Okay, so the next thing is, are stem cells the next big thing in genetic research? So what are stem cells? So stem cells are unspecialized cells that continually reproduce each other. So these are cells, they're just sort of general, and you can, using a few little outside pressures, you can force them to develop into any type of cell. So it could replicate itself normally, or it could differentiate. So remember when we talked about when a sperm fertilizes an egg, at first all the cells are the same. They aren't brain cells, they aren't skin cells. Those are stem cells. And so scientists hope through the study of stem cells that they can learn more about differentiation and how cells become skin cells or brain cells or muscle cells. How do they become differentiated? And through our study of stem cells, we can do. So there are two types of stem cells. One are embryonic stem cells. These are the cells that are found in an embryo that have not yet differentiated. Just like I was talking, a sperm is fertilized in an egg. Those initial cells are embryonic stem cells. All right. These are usually taken from eggs that were fertilized in vitro for IVF, fertility work. But they're not used, so then they're donated to research. All right. There are also adult stem cells. Okay. You have some cells in your body that are stem cells. They're the ones that are used to make your blood and the things that need to be constantly regenerated. They're found in various organs and tissues that need to renew themselves regularly. These can be used to differentiate. They're much more limited. They can't be forced to do as many things as embryonic stem cells. So the research and the use for these is much more limited. And here's an image of the different types of embryonic stem cells or different types of stem cells. Stem cell research is very controversial, but it's also a very active area of research. We've learned a lot about how they can differentiate and turn into different types of cells through our studies in lab. There's several potential applications. So one, we can learn how to control cell differentiation to treat cancer. We can use them to test new drugs. We can use them to replace cells that are caused by cell death, such as Alzheimer's, spinal cord injury, strokes, and diabetes. But there are also huge ethical concerns. And those generally center around the idea of the source of stem cells. A lot of people believe that if you remove the cells from an embryo, whether or not the embryo is formed in a lab, it's destroying human life. And that's a personal issue. Um, this raises the question of about when life began, whether it's ethical to sacrifice life if it's begun, even if it is meant to save another life via research. So there's a lot of controversy surrounding